So remember, there's the central dogma, the way information flows from the DNA to the machinery that are proteins. Why did we ever, so I'm waiting for the microphone to come. Can you actually hear me in the back? Yeah, so maybe we don't need a microphone. Why do we compare shapes? So actually what we want to know, I say proteins are function. What we want to know is how they function, right? Why do we ever bother about looking at the shape? Why do we ever bother about comparing shapes? I briefly talked about that in the last lecture. Any suggestion, any idea? We want to understand what they do. Just a sec, yeah? That's the point. So it's very much shape that determines how the protein functions. Shape influences function. So we hope that from the shape we can learn something about function. And the beauty about shape is it's something very simple. It's a three-dimensional object that can easily measure. I have every single atom in a three-dimensional coordinate space. Once I know the shape, I can define it very well. I can compare it. Function is not as simple. You made a point?
Yeah, the same thing, and also you pointed out last day about the hydrophobic and hydrophilic. So it's like the bond is stronger when it's hydrophobic.
So what she says is, it is true that also shape tells us something about the energy of binding. Shape therefore tells us also something about not only how the molecule looks, but where there are places where more movement will occur. Movement is function. Proteins bind as a dynamical object. So ultimately, because structure is related to function, because we can compare 3D structures relatively easy, three-dimensional objects are easy to compare, and very often or sometimes mostly the similarity in structure leads to a similarity in function. So proteins that have similar structure have similar function. We will talk about that a little bit more. Again, there's one particular protein that I showed here, the alcohol dehydrogenase. This is a view of that three-dimensional structure, and that is this molecule in action. It acts as a homodimer. Homodimer means that two of the same protein coming together, and only when they come together, and in fact in this particular case they are oriented in the opposite way, so this is one and that's the other, and only when they come together do they actually do their function. Thanks, Lothar. Thanks. So since I have it, I will put it on. I don't have to talk that loudly then. How do we get to these shapes? I showed you images. Where do I have them from? Well, from my computer, but that is not the type of question that I'm asking. Yeah? Wait a minute, just a sec, there seems to, the mute is on. Yeah? You were answering?
This number that you take the response, take the domain, the names and the points to where they...
But where do I get them from? Where do I get them from? Who gives it to me? Well, my computer, but this is not what I mean. Yeah? From?
Sometimes scientists will upload the software.
There's a database, the protein database, PDB, and I can download them from the database, PDB. How do they get into the database? Somebody puts them there. Who is this someone? Any idea? Yeah?
I remember, like we were saying, some sequencing machine that was happening in China, so that we need to spend some...
But made a good expression. Yes, sequencing, again, I repeat this because it's just an outstanding reality at this point. A lot of science happens in the Western world, but half of all the sequencing of proteins is done, or of genes is done in China, in one single place in China. So this is outstanding, but this is actually only the sequence, right? So what I showed you here are three-dimensional shapes. Underneath is a sequence. And this lecture is about, can we get from the sequence to the structure with means of computers? So what can we predict? This is, well, how can we use machine learning to predict aspects of structure? But that means that these sequencing machines, they will give us this many sequences for which we want to predict the structures. But where do we get the structures from? Again, they are in PDB. It's true. How do they get into PDB? Yeah? X-ray crystallography is one, so there are experimental methods that put the proteins into the PDB, and X-ray crystallography is one of those. I will talk a little bit more about it, but not much. Ultimately what it is, you take this protein and you make it become a crystal. Now, this is not as unusual as it may sound to you, because the density I told you in the cell is like the density in a solid state. Meaning that some proteins actually sit in a cell and they already look like crystals. In fact, the first protein structures, or some of the first protein structures are such that the people who determined them essentially scooped them out of the cell. Most proteins are not like that. For most proteins you have to do a lot of experimental tricks before you get them to grow a crystal. Once you have a crystal, you actually shoot X-ray beams on it. And from the scattering of the X-ray beams, you determine what residue is where, what atom is where. Now, this is not easy to do, but it's doable. Not easy to do, again, I put a brief tag on that. It used to be, a few years ago, a single structure used to be in the ballpark of $1 million. So making it. And most of that was done by academia, so by researchers. Meaning that the actual price typically, since we are all, as researchers, we are fairly cheap, the actual price is typically higher. So most of that really, there is a lot of machine time in it, but a lot of that really is people working on it. Now the price is down to almost 100,000, yes?
So can all proteins be made into crystals?
So that brings us to the question, for those of you who didn't hear, the question really is, can all proteins be made into a crystal? No. There is, many people have believed that the answer to that question is yes, but I don't believe it's true. So there is a lot of evidence that this is not quite true. One way in which we could approach that question is by asking, well, let's take one organism, such as human, and ask, human has 20,000 proteins. How many of these 20,000 proteins do I know the sequences for? So this is a valid experiment in the sense that there's a lot of pressure to have structures for human proteins, because there's not a single drug developed without having a structure. All drugs target proteins. They are small molecules that always act upon proteins. And before I can develop a drug, I always need to know the three-dimensional structure of that protein that I target with the drug. So drug industry has a lot of money, there's a lot of industry, a lot of interest in it, so there's a high incentive to have many human structures. And there are many other reasons why there is a high incentive to have many human structures. But the answer, how many human structures are there of the 20,000, is at this point fewer than 3,000. Okay? When I now, and that is a technology that I will talk about in one of the over next lecture, we can infer structure through comparing the sequences, and that is essentially the topic that we will talk about today, for about 10 or more than 10,000 others. So that means we are left with sort of 7,000. So we have 13, 20 minus 13, 7. Now, has been a lot of work on these 7,000. And more, some people claim that there are features of some proteins that will elude any experimental structure determination. So some are so weird that they will not be doable. Maybe I'll get to that in a lecture on disorder. But given that I overlook how many vacation days we have, I'm not sure if I'll get. I'll see. Okay. I repeat this view here of 1D strings, such as amino acid sequence, of which we can do many. At this point, we have about 111 million protein sequences known, so for many, many different organisms. There is also the 1D string that is secondary structure assignment. So where is this beta strand here? For instance, in the sequence, it's just a string, so that's 1D. There's 3D, which is ultimately the three-dimensional coordinates of the protein, and then there's 2D. This is a distance map. In this distance map, I simply have a protein with 99 residues. So this is the first residue, and I simply ask my second residue, how distant is it? How close is it? This one is very close, so it's very black. And then the next one, and the next one, and the next one. So we have, for every single pair of the residues in your protein, you have the distance of those. I said that essentially this contains, although it's 2D, it's a dimensional reduction from 3D to 2D, it essentially contains the same information except the mirror image issue. And this is not, so somebody came later and asked, if I took a Rubik cube and I condensed it into 2D, then I lose information. Yes, but this is not what this is. If I took a Rubik cube, then what this would show here is the 27 for every single one of these little cubes. It shows how all the other 26 are close to it, right? And that is not quenching or condensing this cube like that. That contains, if you condensed it, if you quenched it, if you just sat on it, you completely lose the information. But here you have the information, yes?
For the 1D sequence, if you knew how everything interacted with everything else, could you convert it to 1D without losing information?
So the question here is, if I took a 1D sequence, and if I could predict protein structure from sequence, would that then be analogous to 3D, and would I then show that essentially in the 1D string I have the entire information? Yes.
But we just don't know how to do that yet.
We don't know how to do that, and that ultimately summarizes the entire lecture in one sentence. Can we predict protein structure from 1D? For some proteins, under some conditions we can, but for not every single. So get back to the question, can we crystallize every single protein, for instance in human? Can we predict every single protein in human the structure for? No. So the breakthrough is that for some proteins we can do it. And that breakthrough is relatively recent. And that I'll get back to. But most of the time what we will do with machine learning is we will try to predict simplified features, such as where are all these strands and helices. Because these strands and helices already tell a story about function. But let's begin with sequence comparisons, because the first question, once I have 111 million sequences, so 1D strings of the amino acid sequence, the words, so to speak, and the first question that you will be asked whenever you do a new one, so you dig somewhere into the soil, you find a new sequence, and the first question that you will ask is, what is that? What organism is the protein known? Or you look as a clinician or as in a medical environment, you find that some particular protein is involved in a disease. So the first question will be, what is this protein doing? So the first question will be to look for that protein. You may imagine that you can look for that protein through its identifier. But if all you have is a sequence that you find because it's implied in a disease, and this is in fact typically by isolating this protein, all you see is the sequence. Ultimately you can sequence it, so you learn its words, and then you take that word and you want to find out what is known about it. And ultimately the alignment method answers to the question of how similar are proteins and what can I learn from that similarity. Let's just look at an example. So these two, whatever you call it, two peptides here, they're two peptides, OPA and PAPI. And when you see it like that, you immediately see how you can sort of optimize the match. Here, match is defined as simply the same letter, okay, an identical letter. And the simple way to align them, to arrange them such that you have an optimal match between these two words is this way. You can all see that easily. And you can also, even when I tell you this is the best way of doing it, you all will sort of look at it and in your head will try some other permutations. And we'll immediately find out this is the best hit that you can get is 2. Anyway, it's not a big issue because the first letter, the first word here has only 3 letters. If you now came with a new letter, PAPA, then you would immediately see how to put that in. And in fact, this time with the PAPA, you would not sort of look a little bit further, you would immediately see that there is already some PA matching. You would try that first, and that in fact would guide you to the right alignment in this particular case. And then, you know, the story goes on. The point now, you created what people refer to as a multiple alignment. So you align, in this particular case here, 1, 2, 3, 4, 5 peptides. So it's not only a pairwise alignment, it's a 5-mirror alignment. And the question is, what can you learn from it? So in this particular example here, let's just look at the meaning issue. This word, for many of you, is obvious that this is father, that this word here is not father. For some of you, it may become clear that these 3 here are grandfather in 3 different languages. But there is some relation. This is the father. This is something else. Now, what you see here is you have some similarities, some identity, you have some matching, but the matching does not necessarily tell you meaning in this particular case. So we have a relation of words, and these words were chosen such that, in fact, you could imagine that if I align words, in fact, the similarity of grandfather in 3 different languages, this is not coincidental, most likely, right? This evolved from a sort of common ancestor, and they are similar because these languages evolved from each other. There's a relation. But in this alignment problem, you immediately see that 2 things sort of sneak into this grandfather tree that look similar. But in terms of counting, you cannot really distinguish, but they have a very different meaning. Okay? And essentially, this is also going to happen in biology. This is one problem of biology. We are trying to look at this alignment, and from that alignment, guess function. In this particular case, we would say, okay, maybe these 2 here are known experimentally, and since 2 of those here mean grandfather, we would typically in biology then say all of these mean grandfather. Because they all, in terms of aligning them, they all look similar. This is the way alignment methods will work, or multiple alignment methods, and this is the way we will use them to predict aspects of function and structure. And you see it's not always clear. One thing that you absolutely need to define here is an objective function. The function that you will optimize. What was the objective function that we just optimized? Very simple. Is it simple to everybody? Yeah? So the identity of the letters, we simply called a match when we had the same letter, and we optimized simply the number of identities. Okay? In this particular case that we could have also had an objective function that is the percentage of identical matches. This is not necessarily the same thing, mind you. In the case that I showed you, it would have given the same answer. But actually these 2 things give you very often different answers. And that gets me to the sort of longer point. The simplest objective function is the one we tried now. It's a percentage of letters that are identical. Very often, however, it is more complicated to describe a match and to relate a match to function, because some amino acids are more similar to each other than others. I said, for instance, they are hydrophobic residues. And if they're hydrophobic residues, then maybe you would sort of call aligning hydrophobic residues, even if they are not identical, something that is still on the side of good. So you don't match the same letter, but you match the feature, right? And that begins already to show you that there are more complex functions possible. But both of these, whatever you do, they always will ignore the structure. They do not begin by predict the 3-dimensional structure, then compare 3-dimensional structures. They compare sequences. And in sequences, we have some information, and if we could predict, we would have the full, completely 3D information. But since we cannot, this 1D is a simplification, and we, by comparing 1D features, we throw essentially some of the information away that we are looking for. Now, For the time being, let's just look at the string problem, so the 1D problem. And even in the string problem, how do I do this? What did I do on the whiteboard? Essentially, I did two things. In order to align these two sequences here, I first had to find the optimal superposition. And then I had first to define it. I have to inverse this. I have to first define optimal, and then find the optimal superposition. Here are two different. So the goal is to compare these two proteins here, and here are two different solutions. And these two different solutions are back here shown again for this puppy problem. One is local and one is global. What global means, essentially, you are forced to compare these two proteins from end to end. Local means you just cut out something that matches locally. And both for the puppy story and for the protein story, you see that creates different solutions. In particular here, this global issue here would in fact, instead of matching two residues, it would only match one. So it would really reduce the fit. But you require global. Now, what's the interpretation of alignments? The interpretation of alignments, essentially, we talk about evolutionary relations. That's just also what I said about the languages. So evolutionary relations, essentially, we mean that two proteins have similar sequences because they have a similar evolution, meaning that they have a common ancestor. And since they have a common ancestor, they actually have a similar structure. There was some common ancestor so there's the ancestor, two species come off the ancestor, these two species have slightly different sequences, but since in the ancestor they did one thing, and the assumption typically is that in the offspring they do the same thing. They have the same function, they have the same structure, or at least similar structure and function, and a similar role. And often this is referred to with the word homology. So they are homolog, they agree, the words or the meaning agrees. And historically, actually, this comes from Richard Owen, and it comes from this idea that when you look at the whale, that parts of these organs are homologous between different animals. The whale has feet, although they don't look like it. Because the whale was an animal that went into the ocean. So it was a mammal that was on land and then went into the ocean. Now, this view here, sort of the macromolecular view where we compare animals, different species of animals, this is not the world of the proteins. In the world of proteins, we typically refer to homology as that two proteins or two genes have a common ancestor. Okay? And then essentially the sequence comparison reminds us of the evolutionary connection of the organism. Okay, so we compare 1D sequences essentially because we can. We try to find similarities. In 1D sequences we can easily find them, and we hope that we learn about three-dimensional structure and function. Let's begin with the first way of doing that. It's brute force. And brute force is essentially exactly what you did with this puppy problem there. You visually try all possible solutions. That goes back to a paper published by Needleman and Wunsch in 1970, 70, so about 100 years before you were born. The idea simply is the following. You begin with these two here. That's your first step, that's your second step, that's your third step. So you try all possible solutions. To do that visually, in a more systematic way, you write these two sequences here, and then these ones are put wherever you have a match. So the G could be aligning to that G, could be aligning to that G, and that essentially is it. That's the only match, identity match you can get here. For the Q, there's only one identity match. And then you essentially trace through here, build up all the ones, and you trace through in a way that for the global alignment, you know you can essentially only go on the diagonal. You can then that means you have to find the diagonal here and sum up these letters that gives you the best result. Okay? So going from every position, if you start here, you go further, and then the sort of best match wherever you go here will be 3. Okay? So you build up these matches, and then in this particular case what I show here is I take the same number, I sum it up, so this is 1, and this is also 1 because that is still taking that one over, and essentially if I start here and do the alignment like this, I have 1 match. If I start and do the alignment like this, so the second match is here, the QQ, is essentially the GQ. Again, continuing the diagonal, that still is the best match. Now, I could do a little bit better if I allowed for gaps. So allowing for gaps means I allow myself to, instead of always going in the diagonal, sort of go straight at one point. So visually, to align these sequences, I said the GQ, the top match was a 2 here, but if I had gaps, I could actually at least align this E and this L on top. So I could get the overall number of aligned matches to 4 instead of 2 by introducing a gap. The interpretation of a gap in a 3-dimensional structure is that, so you essentially, for instance, have a region in one protein that is like this and folds back like this, and then there may be an issue here, and the second protein here is just like that, but it has this smaller up here. Let's just have the embellishment like this, and this embellishment like this or something. Whatever. What I mean is that these 2 residues that are inserted in one, okay, this is not going to work. So I just have an additional kink in here, and that essentially would be these 2 residues that I insert there. And you already see that I'm not a gifted drawer in any way. Okay, in the dynamic programming, this gap means you move in that direction, so you go off the diagonal. And in this particular case here, this is the way you get at the 4. Now, there's another trace in here that gives you another alignment. And then, in fact, I missed a slide. So now at some point we have to compare the one where I go without a gap to the one where I go with a gap. It means, is it better, so I can say ultimately at the end of the day, if the optimization criterion is the number of matches, then the 4 is always better. If I would take the percentage, then the story may become different, because here you have, well, 1 in 4, and here 4 in 8. Yeah, well, 4 in 8 is still higher. But my point is that you somehow have to find a way to sort of weight gaps, or to put a penalty on introducing gaps. And the simplest way to do that is you simply have a score that has the matches here. So identity of this delta function gives you a 1. If the I is I equals j, the delta is a 1. If I is unequal j, then the delta is 0. So this is simply the sum over the identical letters. And then you subtract the number of gaps times the gap penalty. So this particular case that I showed you here, I have 2 gaps. So n gap would be 2. And then there is a penalty for the gap. And that penalty is a function of a free variable that you have to choose before you do the alignment. Now, when you now look at these alignments for all possible paths, you see typically that 1 stands out, but that there are some sort of local densities, suboptimal paths. And how this matrix looks what are the optimal paths and what are the suboptimal paths, again tells you also something about how simple or how secure the solution is that you have. How many alternatives are there? Are the alternatives all in the vicinity of the one optimal solution, yes or no? And that tells you something about your problem. But let's get back to the gap penalty that I just defined. The way I defined the penalty for this gap was the number of gaps times the penalty. This definition leads to a situation in which these two situations here are identical. Okay? You may see that they look the same because according to the function they are the same. Fair enough. But in this interpretation here, that a gap is something that you add on top, this is more likely than that. Because in this scenario, you add one loop on top. In this scenario, you have a bump, a bump, a bump, a bump, a bump. And in some sense, every additional loop, adding a larger loop, you can imagine that has a functional reason. There's something that's going to bind to that loop. But these little bumps here is less easy to interpret. They seem to be less likely. So what you want is, in fact, when you also look at alignments, and this is a slide from the Hennikovs that introduced the idea of blocks, what you sometimes see, and what I'm showing here, is that protein, the first protein is from the first residue to the last residue. That one, the second protein here, is similar only in this region, from here to here. So mostly for the entire region of the first protein. But the third one is only for the first half. And then you have some proteins here that align only on the second half. And my first lecture I introduced this, or the second lecture I introduced this as an example for domains. So most likely this means that there are two domains here in the protein. But my point is, the function of the gap penalty times number of gaps will not create that. That will create these isolated gaps. So the question is, so this function that is written here will not enforce this type of structure. How could you enforce this type of structure? How could you enforce that when you do these comparisons, so you have to put in a gap penalty, you have to somehow penalize that you introduce gaps, but you want to penalize it such that you sort of have rather the domain-like, so you have rather long gaps in between, one long gap than many short ones. How could you put that into the function? Any idea? How could you change this function here? So again, this is simply summing over the matches and subtracting, in some sense, the gaps, because that is a penalty. But it leads to a situation where I very often have match, not gap, match, gap, match, yeah?
You have a larger penalty for opening a gap, smaller one for continuing a gap?
So the answer is you have what people refer to as a fine gap penalty. So you have two different penalties, one for opening a gap and one for extending it. And the opening a gap is a larger penalty, and the extension of a gap is a smaller penalty. That means to open a gap will cost you more, and you have to set that off by matching more letters in order to get the same score at the end of the day, right? So it still can happen, but it's just going to cost you. So you have to bring in a lot of new hits in order to sort of compensate for that. And typically, in fact, what that is that the opening is typically in the ballpark of 10 times the extension. It costs 10 times as much to open a gap as to extend it. And that clearly creates these structures that we have seen. Dynamic programming, the way I showed you, is optimal, and the final function is simply shown here. So this is the gap open, the gap extension gap. This is just the math of it, is simply counting the gaps. And that ultimately is the entire objective function. You compute that function over the entire alignment. And given this objective function, the simple dynamic programming gives you the optimal solution for aligning these two sequences. That may not be the optimal situation for biology. But if biology is adequately described by this function, meaning you want to have a match of identities, and you sort of can find the penalty right, then the algorithm is completely giving you the optimal solution for that. that brings us to this question, what is better, global or local? And is really, in fact, the number of matches the best criterion? In the function I just summed up over the number of matches. I did not have a percentage. So what do you believe? First of all, is it global or local? So is it this one here, the green one, where you have two right ones in an alignment of 2, versus the purple one here, where you have 8, so you have 4 matches in 8. 1, 2, yes, exactly. So what is likely going to be better? 2 and 2 right, 100%. 4 and 8, 50%. Which of these two alignments do you believe is better? The question is, how can you answer that? Well, you can Google. This is not quite the answer that I meant. How can you think about finding a way for an answer? Yes?
If the number of matches is the best criteria, then the values we see can go to number of matches.
That is a very correct statement. But I'm asking, how can you think about a project if that would be your master thesis? How could you try to find out whether it's one or the other? Again, assume that you were the first so that you cannot Google. Today you can Google the answer. It's very, very simple. There's a clear answer to that situation. But what would you do? What would you look at? Okay, there was somebody, I don't know, was somebody here? Proportion. Yeah, well, the proportion essentially is you compute the percentage, right? And my question is, how do you know whether 2 and 2, which is 100%, or 4 and 8, 4 and 7? So I keep having issues to count. Well, actually, this is both wrong. I'm wrong and the slide is wrong. I have to correct the slide. This is 9. Whichever you count the shorter or you count the longer. It's 4 and 7 of the shorter one, and 4 and 9 of the longer one. So that gives a percentage. But my question really is, how could you answer which one of those is the biologically more meaningful situation? Yes?
Maybe based on like the database they have. So like, the question is, what do I need to do with the match they have? And then
that's a great answer. So unfortunately it's a very complex answer. So what he says is that I cannot answer this question before I don't know what to do with this. So my question really should be, what is my goal when I compare this? And then I will ask, with which of these two answers do I satisfy that goal better? And I really like this way of thinking. That the problem is to find out what your goal is. Ultimately, the goal is to find similarity in structure and to find similarity in function. And that will take a little bit longer. It's a little bit more complex. There's a simpler way to answer that. Yes?
Maybe we could compare your respective 2D and 3D structure and look if they are similar.
This is ultimately going in that direction. So we could look at a bunch of pairs of proteins for which we have similar 3D structures and see whether these two things are similar. What I meant is simpler in this particular case here. If we look at the GQ, there's two things, okay? And you look at the matches of 111 million proteins, you will find that again and again and again. You will find it so often that you will realize this is a totally spurious or random hit. If you have 1100 million sequences, each of those 400 residues long roughly, finding a GQ is completely random event. Okay? So you can already answer from mere statistics that this is not meaningful without knowing anything about structure and function. So while your way of thinking is right, and the way of in fact going and looking at the 3D structures, and we will do that very soon for something else, that answer. Here we can approach this problem from a much simpler perspective. So always try to find the simplest possible solution. And for this particular case, you can trivially make out that this is wrong. This one is not right by that, but you could try. So if you have 9 MERs, how often can you find in the database 9 MERs that are unreasonable? And then you have to actually begin to look at structures, and then you have to actually begin at the question, for what purpose? So this is a little bit more complex, but it's already clear that this is better than that. So that brings us to the next issue. So far we have always talked about identity, so we have matched, the match criterion was always, we have the same amino acid. I already said that biochemically, maybe there are features that are important. So what I show here is a sequence of 2 potassium channels in 2 different species. And in fact, the part that is the potassium, that is sort of the gating mechanism of the potassium channel, that lets potassium go through that channel, are the ones in yellow. This alignment that you see here, in fact, was the beginning of what became a Nobel Prize for McKinnon at Rockefeller University, because he actually found that here in this alignment, these 2 proteins are similar. If they are similar, then he could use the one that is not the human sequence here for doing the structure determination, because the human protein in this particular case he didn't get the structure for. For the other one he did, and that was ultimately a long path that led to a Nobel Prize. But again, my point is that these 3 residues here, they do the same thing in 2 different organisms, although, as you see, only one of them has the same letter there. 2 of the 3 have different letters, but they are the sites that regulate whether a potassium is let through the channel or not. Okay, so they are the there's a name for that, it escapes me. The sensing, or I can't remember how this 3 letters are called. But anyway, so this is an example that tells you we need more than identity. We need to go for something that considers the biochemical similarities. One way in which we could do that is we could say, okay, there are positive charges, there are negative charges, there's hydrophobicity, there's hydrophilicity, there's polar changes, there are size of residues. We put all of this into some vector that describes the biophysical features, and then we define the similarity between biophysical features. This is one way in which you could do that, and people have done that. Another way in which we can do that is the path that is most likely much closer to your guys' background. Was done by Stephen Hanikoff and Jorge Hanikoff, and led to something that is called the Blossom matrix. So essentially what they did is they had a bunch of proteins that they knew have similar structures. So essentially, in some sense, they began from what the 2 of you mentioned. So they picked a bunch of things that have similar structures or similar function, or at least believed to have similar function. And then they asked for the bunch of things that are similar in their function and structure. What are the changes that we see? Simply count the log odds. So initially, they had a few thousands of pairs of sequences of things that they believe do the same thing. And then, essentially, the statement is, wherever the letter is the same, that is important. But wherever in these cases the letter is different, that may also be important because they do the same thing. So we compile a simple log odds matrix, shown here. And then what you get, sorry for the quality, is the 20 amino acids. And the 20 amino acids again here, they're somehow grouped. And that's diagonal. And the diagonal essentially means what do you get as a positive score if you match the identical letter. And you see here for the C, that gives you a 12. For the W here, it gives you 17. But let's look at a few examples here. For the AA and the EE, it gives 1 gives 2, 1 gives 4, and 1 here, WW, gives 17. Meaning that for some identity matches, it's more important to have them, the WW is more important to match W, than it is to match an A, okay? This is what this matrix says. And it continues saying something else that is actually maybe somehow counterintuitive, but that is sort of following up from what I said before. The match of the L here on the M gives the score 4, while wait a minute, that is an A on an A gives only 2. That means the identity match for an A, happens to be an alanine, is less important than matching 2 two hydrophobic residues here, the L and the M. And there is another highlight here. Some are negative. Here, the I on the A is a -1. And then some of these are, in some cases here, the number is much more negative, so matching W onto a C is -8. Anyway, so this matrix now reflects the biochemistry, and you can simply plug that matrix into the algorithm that we said, that I showed you before. You simply replace the delta function by this lookup score, okay? And this, in fact, is the most used current matrix here for the comparison of protein sequences. By the way, there's a nice tool developed by Francisco Melo that allows you to sort of play around with dynamic programming methods, yeah?
You were saying that the score index is an index of importance, but what do you mean by important? Does it mean that they usually, like for example, like the case for the W and W, does that mean that if always W shows with the W, that means if it never shows with another character, that means it should be long or something?
I made a mistake, I should have the function again here. So the question is, what does it mean is the most important? Does it mean that W has the highest number here, the W should always match W full stop? Or something. What does it mean? And ultimately the answer to that is unfortunately a few slides before. That's why I said I'm sorry for not having the slide here. And this is also not the last full Smith-Waterman score. Where's the full Smith-Waterman score? It's also not I'm looking for the formula. Where was it? Where did I have the full Smith-Waterman score? Here it is. So this is the entire formula for finding the optimal superposition between two proteins. And now this is what you change. So the M used to be, if W is W, it gets a 1. If A is A, it gets also a 1. If A is W, it gets a 0. And that now is replaced by the matrix that I showed you. Okay? And then does that now mean that the W has to always match the W? No. You will optimize that function. And in some cases that will, in fact, since the W has a 17 there and the alanine has a 2, so matching W is much more enforced, so it brings you much more brownie points. But you can find a path. If the path leads to a lot of alanine matching instead of one W, that could be the better path.
Yeah, my question though was that when I look at the matrix, the index matrix, I understand nothing. So when I see W matches W with 17, I don't really know what that means. Why W matches W with 17 and why A matches A with 2, for example. So that's more my...
Sorry, I misunderstood your question. So the question, I believe, was how did that matrix come about? And that matrix came from essentially here the log odds. You simply look at a bunch of pair comparisons. You take a bunch of proteins for which you know they do the same thing or have a similar structure, and then you simply count at each position. How often do you see S on S? How often do you see W, W? How often do you see? And you translate that through log odds, and that's exactly what you get. Those are the log odds. That is the formula that I show here. Okay? Is that the answer?
Yes, just to confirm. So that the fact that we have W with W here is 17 means that W almost all of the samples showed with only W. And it did not show.
So the question here is, does that then mean, the W17, does it mean that in all these comparisons of protein sequences, the W matched on W and essentially on nothing else? Exactly. That's exactly what it means. Yes. Because you assume by comparing a bunch of sequences that you know have similar structure, that what you see as the pattern of, let's call it conservation, or the pattern of match or mismatch, is exactly a neutral, meaning neutral in the sense of not changing structure, not changing function. And that means that what you see here is reflecting, if you sort of reproduce that, then you have things that look like the things you know are similar. Okay?
So this is basically weighting the matching. There are matching that are more important than all
Yes.
based on this.
Yes, that's exactly what it is. So some matches, if you want, this is Orwell once again. So some things are more important to match than others. But again, another point of this is here, some of those are such that even mismatches, so some, particularly not every mismatch is a mismatch. So the M on the L here is a mismatch that gives you a lot of points, because you match, in fact, a biophysical feature. And this matrix here also gauges the degree, if you want, this is a simple way to gauge the similarity of biophysical features that you take out of the data. You have no assumption in here about you understanding what is charge, what is energy, what is hydrophobicity. You simply count for objects that you know have similar features, structure. In those days, actually, it was not structured, but in principle, this later has been redone with exactly your idea of taking a bunch of proteins that have a similar structure. Yeah?
They also have a lot of similar functionality, or like proteins having similar functionality as well, as to be able to be sure that the matrix doesn't vary different functionalities, right?
I did not understand the word before, the two words before right. So the assumption really is that this is the result of pairs of proteins that have similar function. Was that what you meant?
How can we ensure that this matrix doesn't vary different functionalities or different functionalities?
So again, be careful. When I align two proteins by that matrix, I cannot be sure that they do not have different function. The logic goes the other way around. If I take a bunch of pairs of proteins that have a similar function, okay, then statistically, if I have enough, there will be exceptions. There will be cases where there are some exceptions. But overall, this matrix, this is determined by where the mass of the data sits. And overall, the things where change is allowed is where change does not affect function. Otherwise, these things wouldn't have the same function. Okay? So this is a very simplified condensation of the information of a bunch of pairs of proteins with similar functions. It's the simplest thing you can do. It's not very intelligent, but essentially you take 1 million pairs of proteins. Again, in those days they took only many thousands of pairs of proteins, but today you can take 1 million pairs and can condense that information into 20 by 20, or this is symmetric. So not 20 by 20, but less than 400 numbers. So it's a very, very simplified view. And that cannot guarantee that what you find applying it is really the right thing. But maybe if you use a simplified strategy, this is the best way of doing it. Certainly is clearly better than using identity. Yeah?
So that this basically means that as and when we discover more things that are similar structures, this matrix could eventually be.
So over the time, we ought to readopt this matrix and redo this. It would change and it would become better. Now, comes something, I'm not entirely sure how to call that. This matrix here has been done a long time ago. You may see that the publication date here is 92. And this is still sort of the most frequently used matrix, although people have redone it with better data. And ultimately, I believe the new approaches have not been taken over because there are too many groups out there who have done exactly this but have made mistakes. Incidentally, this is also one of those interesting stories here. No, I have to keep the paper. So this PNAS paper had a mistake in a formula, which was discovered at some point, I can't remember, 5-6 years ago. And this mistake led to a slightly different solution. So the matrix that was actually had been used for almost 20 years by everybody was wrong. They corrected it And it got worse. So this is one of those stories where there is some explanation why that is the case, but it's way too complex. Conclusion, in computational biology it pays off to make mistakes. Okay, so there are new matrices, and for some applications new matrices are used, but overall this relatively old one is still very popular. Most likely because it's very simple. It's the simplest thing you can do in some sense. Now here's another issue. So the dynamic programming now with this matrix gives you a biologically meaningful solution, and it gives you the optimal solution. But it has a problem. The problem is time. The complexity of the algorithm is in the square of the length of the two proteins. Okay? So why would that be a problem? So these protein guys are 400 residues long, 400 times 400 is a small number. Why should this be an issue? Anybody? What's the problem?
Memory?
400 times 400? Your telephone can do that. Whatever telephone you have can do it. Well, is your telephone older than 20 years? But most likely any telephone can do that in the room. I don't know, maybe some of you have now shoelaces who can do that. That can do that. So that really is not the issue, yeah?
If you have something new and you want to compare it to the database, and if the database isn't 400 long, it's...
So the point is you have to do it again and again and again. And so that you have to do comparisons against entire databases, and ultimately the databases, they only double, but the comparisons of databases, this increases over 2 years over 20-fold, the number of comparisons that you have to make. And that is where the problem is. So it begins by simply, you find this one new protein, and you have to run it now against 111 million times 400. And then suddenly this becomes an issue. There's another issue here that has to do with how to choose the parameters, the free parameters. We have the get penalty issue, and assuming that the matrix is the right one, but the matrix essentially is also an open issue. So how can we speed up? I'm sorry that I started late. I still like to wrap up with this idea of speeding up. Say it again? Parallel. Oh, so I actually don't really know if I took a 400 residue protein and compared it against 111 million with the typical dynamic programming. So most likely that would be in the ballpark of days. And now if you had 10 processors or 100 processors, you would break it down to a few hours. We get 1,000 queries every day. So then suddenly even that doesn't work anymore, right? So yes, parallelization is one attempt, but it's not enough. Could be one attempt, but it's actually not enough at all. Yes?
Try a different approach, like a class, and do this algorithm. I mean, again, stochastic, just pick where the sequences after the class.
Yes, so the answer is, I'm going to explain it to you, I'm not going to repeat what he said, because ultimately, the way I understand him, he has the solution that I have anyway on the slides. The idea to the solution, you begin not aligning entire matches, you look at stretches, at words. You pick words, you randomly pick some words in your first sequence here, and you simply look for identity. Say the word size is 3. 6. YKL is the first word match that you have, and that's the second, third, and fourth, and fifth. So if you look now at word seeds of 3, this is all the matches you can get. Yes?
How do we decide the class size?
So what is the word size? Is it 3? Is it 10? Ultimately, the answer to that question is 1 doesn't work. because then we are back to dynamic programming. So you begin by 2, but then you see for 2, and that brings us back to this example that I had, even 10 or 20 years ago when this, a little bit more than 30 years ago when this method was shown, 2 was already not enough. They found spurious hits. So number 3 was the first number where they, those days, did not find anything that was nonsense. So they started with 3. That's the way they started. Today, the database is so large that 3 gives you problems, and people actually have moved to 5. That's as simple as it is. So you begin with something that if you find that, at least you can be sure that this is not a totally random hit. That's how you start. Yeah?
Just a question. So like you said, over time the database size increases. Do we change the previous ones? So like the previous ones, do they change as well?
So the question is here, I said that the database is increasing, so we are now at 111 million. Do previous records change? Sometimes yes, because sometimes people find out that there were mistakes in the sequences before, but that is the exception. Mostly there are just new ones added, at least on the level of sequence. For annotation it's slightly different. So the next step then is you try to extend around this local word match. So you have a local word match, and you essentially try to see, can you extend the alignment in either of those directions? How would you extend? What algorithm could you use to extend the alignment, to make it a little bit longer? Simply to ask, is it the match, is that only a match here, or is there also a match a little bit around it? Is it only 3 or is it more than 3? How could you do that? Any idea? Yeah? Yes, so you can essentially do exactly what we said before. In between, to extend, you can use the dynamic programming. Why now is this not a problem? Or why do you still, if you go back to using dynamic programming, why not use the dynamic programming from the beginning then? The point essentially is in the numbers here. I said that dynamic programming is the speed of the dynamic program is the length squared of the 2. The length squared of the entire thing is a huge number. The length squared of 3 plus 2 is a very small number. So by breaking the problem down, you square the breakdown of the speed that you increase. Just if you had only 2 fragments, okay, you already have half the CPU for running this dynamic programming on the 2 halves. It's 4 times faster already. In this example here, you see that we have a breakdown to 5 parts. So the speed is improved by much more than the factor of 4. By a simple procedure. And yet in between, you have what you had before, the optimal solution. There is some issue that has to do with what do you do in between. And ultimately the answer of this method as it stands here is nothing. You keep it open. You don't know. You actually can simply by the method not say it. Maybe there is no similarity. Maybe this is a long loop. Maybe it's an additional domain. We don't know. Okay? And then we get into why can we do this? We can essentially only do that because we have to work out the statistics. And I'm still going to say it, although we're getting late in time. That's going to be the last point I want to make. The major problem that now becomes to be an issue is in the statistics. So what's the probability of matching a word? That brings us back to this question here. How do I start? Why do I choose 3? Why do I choose 2 or 3 or 4 or 5? And ultimately the answer to that is statistics. So for whatever number you choose, you compile a background distribution. So you essentially take a tumor, run it against the entire database, see what the score is, and then you hope that the story that you have, that the hits you have, are outliers. If they're not outliers, if they're right in the distribution, they are meaningless. This would be the example of saying, I cannot start with 2. Because 2 gives you something that's right here in this distribution, or even here, but it's still sort of shadowed by the background distribution that I would call random matches. And random matches, they may be meaningful. So the match between these two proteins may in fact reveal something about function, but I cannot tell. Yeah?
This way, how do we find like special properties of proteins? Like if not many proteins possess that property?
Well, these, if not many proteins possess these, then these not many proteins would stand out from the background. And the ones that stand out would share something that all the others don't have. This is exactly what the algorithm is sort of focusing on, or the idea of statistics is focusing on. Because since the ones that have, so you could look for similarities in them, or you could have a bunch of things that are completely different from each other, but since they all differ from the majority, they stand out. You would not see the similarity in those. That would be the end of that. So you cannot find, if you asked, how can I find a sequence similarity between two proteins for which I know they have a three-dimensional structure the same, but they have very different sequences? And the answer is, we cannot. Sorry, that is a second interpretation of your question. That's what you meant, right? Yeah, in answering I sort of realized that I'm going in the wrong direction. So that you cannot find out. Okay? But again, the statistical importance here is you need a score that assesses how far away your current hit. So you have a word of 3, a word of 5, match score, and the match score again comes from this summing over the Blossom matrix that I showed you, over 17 for the WW, the 2 for the alanine, subtracting the gaps. That is the score that I plot here, and then you show, or you look how far the score that you actually have of something that you believe is a meaningful score from randomly running anything in the database. And the expectation value, the significance of this, is given by 2 values, or by 3 things. Now what you have to look at is, what is the mean, what is the standard deviation, and how far away is your score from all of that, given the mean and the standard deviation. Ultimately, how far are you away from the bulk of the background distribution? That's what you measure by the expectation value, and as long as the expectation value is far away from the distribution, you can say that the hit that you have has not arisen by chance. Or you can define the probability that it has not arisen by chance. If you have an e-value of 10 minus 3, that means that the probability that it has arisen by chance is 1 in 1,000. With a database of 111 million, that actually is a small number. So you want a probability, an e-value of 10 minus 10, or something like that. Anyway, this is where we stop it here today. Thank you for your attention. Are there any questions? Thank you.